of the seminar. Okay, dear colleagues, today our seminar and our lecturer, Professor Jeffrey Bode. He is from Zurich and he studied chemistry and philosophy at Trinity University in San Antonio, Texas. After PhD studies at the California Institute of Technology in Zurich with Professor Eric Carreira, he spent 2 years in Japan as a postdoctoral fellow with Professor Keisuke Suzuki at the Tokyo Institute of Technology. In 2003, began his independent career in the United States at Santa Barbara and then the University of Pennsylvania. In 2010, he moved to Swiss Federal Institute of Technology, ETH, in Zurich as a full professor in the Laboratory of Organic Chemistry. Since 2013, he is also a principal investigator at the External Institute of Transformative Biomolecules at Nagoya University, where the Bode Group has a satellite laboratory. Starting his academic career, Jeffrey's research and teaching have been recognized by numerous awards, including an Arthur Cope Scholar Award and Elias Corey Award for Outstanding Original Contribution Organic Synthesis by Young Investigator. His research group founded and operates the Make a Molecule Out Research Program for Young Scientists. He has served as a chair of the editorial board for Organic and Biomolecular Chemistry, co-editor-in-chief of Helvetica Chimica Acta, and is currently an executive editor for Encyclopedia of Reagents for Organic Synthesis. Research in his group has led to the formation of 2 companies. He currently serves on the Division 2nd Research Council for the Swiss National Scientific Foundation and is the Director of Studies for the New Biochemistry and Chemical Biology curriculum at ETH Zurich. Dear Jeffrey, please, you can start your presentation.
Great. Thank you, Valentin, for the very kind introduction and also the invitation to present today. It's really an honor to have the opportunity to speak to all of you, and I'm also very honored to see so many people who come and join a chemistry seminar during this time. In the last months, we've gotten to know, many Ukrainian students who've come to Switzerland, and I think we're all so impressed and so inspired by all of you who continue to work and study and do research and keep a good spirit through what must be absolutely terrible circumstances. It's really an inspiration to all of us. I feel almost embarrassed to come talk about my chemistry today, but I hope can provide a little distraction from everything else going on. So as Valentin said, I did my PhD studies and my postdoctoral studies in synthetic organic chemistry. And in classical total synthesis, I think one of the most interesting or attractive aspects is we have a target compound, but we don't have a map of how we should make it. So it provides an opportunity for a lot of flexibility, a lot of creativity about how we put a molecule together. So if we look at a famous example of a complex molecule made by many groups like Taxol, we can see that there are many different solutions to the synthesis of it. So each group has a different starting material, each group has a different route, and a lot of freedom about how we go from the starting material to the final product. And I think as a student, that freedom and that flexibility is very exciting. You get a chance really to think differently, maybe than others, about how you would make a molecule. But as we get older, or if we're working in drug discovery, we sometimes don't want so many questions about how to make a molecule. We would like to know very quickly and very reliably how we can produce a target compound. And my group now works a lot in peptides, proteins, chemical biology, and synthesizing peptides, which are actually quite complicated molecules, is very different because we have predefined building blocks, we have predefined routes and chemistry, so that we can look at a target molecule like a peptide, and we can immediately say, okay, I can make those from these building blocks. These building blocks are commercially available. Actually, they're quite inexpensive nowadays. I might need some special resin, but I can also buy that. And I know exactly what steps I need to do and in exactly which order I need to do them in order to produce a target molecule. And I think that this is it's an interesting compromise. So in peptide chemistry, we can't make everything, but if we can write down a peptide and the building blocks are available, we can be almost 100% sure that we will be able to make that compound. And also, since we know exactly which reactions we need to do, we can automate them. So there are many peptide synthesizers. I think our lab has 5 or 6 of them. We know how they work, and we can use those to rapidly make lots of different peptides. And so what we're doing in peptide chemistry is we are trading our ability to make anything for our ability to make many things quickly and reliably. And I would argue that this is actually the approach that nature has taken as well. So in biological systems, most of the chemistry or much of the chemistry is done by proteins and enzymes. And all of these proteins and enzymes are made from 20 different building blocks and one single reaction. Ribosomal amide bond formation is the only reaction that is used to make thousands of different proteins with many different properties as catalysts, as scaffolds, as signaling molecules. Basically, almost anything you can imagine you can build by using just these 20 different building blocks and a single amide bond forming reaction. So we've been interested in this for a long time, and we have been thinking about how do we maybe get a little more structural diversity, a little more interesting chemistry than what you would get out of a peptide, but still using this concept of having predefined building blocks and automated reactions so that we can produce drug-like molecules reliably and knowing exactly how to make them. So even from some of these more complicated molecules like this, we can divide them into very specific building blocks, and we know exactly what reactions we need and exactly which order we want to do them. And if we know that, then we can start to think about automation, and we can try to build machines that allow us to put these predefined molecules together in an automated fashion. And so this is our goal, and this is what I will talk about today, is our approach to producing drug-like molecules. By taking small predefined building blocks, we can build virtual libraries. So with millions of compounds of molecules that we would like to make, and then be able to select those molecules and use automated approaches. Either I'll talk about these capsule-based machines and also some plate-based chemistry that we've developed as well, for then producing these molecules and being able to then use them for biological studies, bioassays, or for determining the properties of these compounds, and using that information to go back and make a cycle of how we design, make, and test those compounds. So the technology that we are using for much of the automated synthesis came out of our laboratory and is now a small spin-off company from our group called SimpleChem. Simple was started by a former PhD student in our group, Dr. Benedict Vonner, and has been joined from some other group members and collaborators, and there's a few more employees as well who are not shown on this slide. And the concept of SimpleChem is really to be able to completely automate the production of organic molecules in this small, reliable approach. I'll go through how it works and the type of chemistry that it can do. It certainly can't do everything, but it can do quite a bit. And from the reactions it can do, we can really make a lot of molecules in a predictable fashion. So our approach to automation was to first think about what is involved in a chemical reaction. What do we need, right? And basically, we need just a few things. We need some starting materials. So if we're doing, for example, a reductive amination, we need an amine, we need a carbonyl like a ketone, and we need a reducing agent. We need a recipe for doing that. So we need to know the order of steps, we need to know the solvents, we need to know the temperature, we need to know something about the reaction workup. And that's typically expressed in a text-based fashion, such as you would find in supporting information. And then for the actual conducting the reaction, we don't need so many things, right? So we need to transfer reagents, we need to stir or mix reagents. Perhaps we need to heat or cool a reaction. And a lot of the time in organic chemistry, we just need to wait. We just need to leave the reaction alone and wait for it to proceed. And so we thought that if we break a reaction down to these elements, then we can build a console. So we can build a hardware that has these basic functions like pumping, mixing, heating, and waiting. We can build capsules, which would contain the reagents that we need for the reactions we want to do, for the reaction workup, and possibly for the purification as well. And then we need a script, basically a little program that tells the machine how to work and when to work in order to do a reaction. And the reaction that we wanted to automate at the beginning was some chemistry that came out of our laboratory starting about 7 or 8 years ago. And that's called SNAP chemistry, for tin amine protocol reagents. So this was chemistry that we developed over the course of several years for the synthesis of saturated and heterocycles. So saturated and heterocycles are probably the most desirable class of molecules for drug development. They come in many different flavors. So we have morpholines, diamorpholines, piperazines, we have substituted versions. We can have larger or smaller ring sizes like oxazopanes. And what we developed was these SNAP reagents with a tin moiety and a free amine. These condense with an aldehyde starting material to form this imine. And then in the presence of a copper promoter or catalyst, we can form this carbon-carbon bond and produce directly the unsaturated heterocycle. So these reagents are nowadays quite popular. You can buy many of them commercially, and they're used by hundreds of pharmaceutical and biotech companies for producing molecules like this. And what is particularly nice about SNAP chemistry is that it tolerates lots of different groups here. So these can be aromatics, these can be heteroaromatics, it can be aliphatic compounds. You can have quite a few unprotected functional groups. And with pretty much just one standard set of reaction conditions, not everything, but most of the reactions work quite reliably. So these are the good points of the SNAP chemistry. The negative points is that although the reaction does work quite well, it is a bit labor intensive. We need to preform the imine, we need to pre-make the catalyst by mixing copper and lutein. And then the workup of the reaction is quite involved. We need aqueous extractions, we need column chromatography. We need to worry sometimes about the quality of the reagents, particularly the copper. The batch of the copper can affect the reaction, although nowadays we have pretty good sources for this. And the tin reagents themselves, although they're fine to handle in the laboratory, you should always take care if you're working with organotin compounds. And so we wanted to minimize exposure to the tin reagents while still accessing the very nice chemistry that they allow. So over a course of several years in our group, Benedict and others built an automated approach to doing SNAP chemistry. And in the end, we came up with these capsules that have 4 different compartments, so we can change the reagents that are in the different capsules. And so if we build a capsule for doing SNAP chemistry, we have one component which is containing an immobilized SNAP reagent. So what we do here is we use, we start from an azide and load it onto a solid supported version of triphenylphosphine. And then this will react with an aldehyde that we add to do a Staudinger type reaction to give us the imine. And it will release the SNAP reagent from the resin and form the imine in solution. Once the imine is formed, we can tell the machine to pump this over the second component of the capsule, which contains a copper catalyst and an elutadine ligand. And by passing it over this column, we can affect the cyclization to give us the saturated and heterocycle, along with some impurities. So we still have the copper species that's present, we have the tin that is released, and we might have some other impurities such as the unreacted aldehyde that are still present. So we can then take this mixture and pass it over a column containing a solid supported acid, so SCX. This captures our desired saturated and heterocycle, which is an amine. The other components will pass through and go to the waste. And then finally, we can release that and basically elute the product as a methanol solution. And all of this chemistry is done. So we have this capsule and then just a little script, which is built into the machine that tells us exactly, or tells the machine exactly how to do SNAP chemistry. And essentially it's just a little program like this, which uses only 3 elementary operations. So transferring or pumping, stirring, or heating. So the step, for example, for cyclization uses heat to accelerate the reaction. And that's it. So in the end, our chemistry is reduced to this capsule and this little script. And that's it. So this is one of my PhD students, Angus, showing us how this works. So what we do is we start from an aldehyde. So there's an aldehyde that goes into this vial. We have a completely automated system. There is an RFID chip in the capsule. So we scan this and now the machine knows exactly which reaction it's doing and which flavor of SNAP chemistry it's running. And then you just press start and you can then leave it for the next few hours while it does its chemistry. And once you've programmed the machine and optimized this, it really works quite well. So we can take the different flavors of SNAP reagents. So we take a different capsule for each type and a different aldehyde. And this is all data that is done completely automated reactions using this platform. And so as you can see, it tolerates quite a few different types of aldehydes, and we can have different types of heterocycle forming capsules to produce standard things like morpholines and piperazines, and also some more exotic things like these spirocycles or benzoxazopanes. The chemistry is quite clean. It's not perfect, but you can see that this is a unpurified, saturated and heterocycle directly off of the machine. This is the product here by LCMS. This is a 1H NMR. You can see a little bit of impurity here, which is a little bit of residual tin reagent, but overall it's quite clean, just directly off of the machine. And this is also very reproducible. One of the things we wanted to make sure is that if we do this again and again, that we see similar results. And so for a panel of different saturated and heterocycles where we ran the same reaction multiple times, we see comparable results each time. And we use the same principle. So some work needs to go into optimizing and training the machine, writing the program and working out the compositions of the capsules. But once we do that, we can provide capsules that are really quite reliable for different reactions. So we can do things like the SNAP chemistry, which I mentioned, but also Mitsunobu reactions, amide bond formation, reductive amination, and along with some kind of more standard but necessary chemistries like Bach protection or Bach D protection, or other types of protecting groups. And just recently, we've introduced capsules for Suzuki couplings and azide formation and a few other reactions like this. So now that we have a few reactions, so we don't have many, but you actually really don't need all that many. So we can look at the chemistry that we have and think about, okay, what building blocks are available and how can we use these automated reactions to synthesize libraries of compounds by using 2 or 3 fully automated chemical steps. So we start from a few of these capsules and some of our standard building blocks. So these come actually from enamines libraries. So we have aldehydes, carboxylic acids, amines, along with some more specialized reagents like SNAP reagents or diamines. And then by sequencing reactions together, for example, by reductive amination, followed by Bach deprotection, and then another amide bond formation, we can prepare virtual libraries of really millions or even billions of different compounds. And so we can, using enamines database of their commercially available starting materials, along with the sequence of reactions that we can do, we can build up these virtual libraries that cover quite a broad range of chemical space and look really quite interesting and drug-like molecules. Here's just a few examples out of the library that you could in principle make by these automated reactions from readily available starting materials. So for actually doing this, we typically work with a little bit smaller data sets. The one I showed on the previous slide is something like 17 billion compounds. That's way too many to handle. So typically what we would do is make a restricted set of building blocks and build a kind of focused library for this. So for example, we developed what we call iSNAP chemistry, in which we form a saturated and heterocycle using an aldehyde as an input, and then release a protected ketone. And then we can use this protected ketone for a reductive amination using a commercially available amine reagent. So if we take even a relatively modest number of starting materials, we take, just in this case, 4 of our iSNAP resins. We take about 170 amines and about 200 aldehydes. Then we can combine those together and using the automated reactions. Then we filter those based on their molecular properties. So for example, we want to remove ones that are too big or that don't look like they're going to be very good drugs through a variety of different metrics, and basically reduce that down to a few million compounds, so maybe about 1 to 2 million compounds, and then define this as our virtual library. And once we have this virtual library, we can work with partners, so either academic partners or commercial partners, who take the library of virtual compounds and conduct in silico screening. So for example, we have done this with BASF, who offers what they call an open innovation platform. You can submit to them libraries of compounds, and they screen this in a virtual fashion for a variety of different targets. These are for agricultural applications. And so we did this. We sent them about, I think, 1.2 or 1.5 million compounds, a virtual library. And they came back actually quite excited with a number of different, actually it was several hundred molecules that they were interested in that showed promising activity in the in silico screen. And so we took about 40 of those compounds and then used the automated synthesis so for preparing these compounds. So this was all done by Angus, basically as a hobby. He has another project, but because he could use these automated instruments, he was able to produce all of these different molecules, and then using preparative HPLC, he could purify them and even in most cases separate the diastereomer. And this produces quite a bit of compound. Even working on relatively modest scale, we're able to isolate 10 to 20 milligrams of all of these different molecules and the corresponding diastereomers. So we continue, so Simple continues to develop new reactions and different ways of combining them together to produce these kind of drug-like molecules. But as an academic back in our research group, one of the things that we were thinking about a lot is that although this is really good chemistry, it makes beautiful looking molecules, we still have this very high synthetic overhead. So in order to access these kind of structures, we need these preformed iSNAP reagents. We can make them and they can be scaled up. We've made many of these on 10, 20 gram scales, but they're not commercially available. So you have to make them, or you have to ask someone to make them for you. The SNAP reagents are more widely available. There's quite a few vendors that will produce them. And also Simple sells capsules for the most common ones, so about 10 or 15 of the most common ones. But there's still not all that many available. So you either have to make them yourself or you're limited to a handful of compounds. In contrast, if you look at more common building blocks or more common functional groups like aldehydes, carboxylic acids, amines, boronic acids, now there are thousands or even in some cases like tens of thousands of different compounds available. So in particular, enamine has done an amazing job of making things like amines and aldehydes, and carboxylic acids very widely available. So you can purchase really hundreds of them and have delivery within a few days. So we wanted to think about, how could we access some of these, saturated and heterocycles, really, attractive molecules for drug development, but using the vast libraries of these more common building blocks that are available because of companies like enamine. And so to make a long story short, I won't go through all the development. We developed an assembly reaction also using a tin reagent, a radical-based approach, in which we can start from a commercially available aldehyde or ketone. We condense this with this sulfonamine, and we form this imine intermediate, and then a radical-based addition gives us this product here, and then we can remove the sulfonyl group from the amine in order to give us what we call SLAP reagents. So these are similar to our tin reagents, but have a silicon here instead of an amine. And the advantage of this is that we are able to basically just start from any commercially available aldehyde or ketone, and in 2 steps we can produce these kind of SLAP reagents. And we took some time, so Moritz and Ice, 2 graduate students in the group, took some time to optimize these procedures and to work out an automated workflow for preparing these kind of SLAP reagents. And to explain how this works, let's look at the mechanism of what we call SNAP chemistry and SLAP chemistry. So the SNAP chemistry, which we introduced before, uses copper as a promoter. And what happens is the copper-2 oxidizes the carbon-tin bond to form this C-centered radical. The C-centered radical undergoes a cyclization to form the saturated N heterocycle, and initially delivers this more stable N-centered radical. And then the copper-1 that is formed in the first step reduces the N-centered radical to go back to copper-2 and to deliver the product. So this is 2 single electron transfer reactions, so a single electron oxidation and a single electron reduction. And this is exactly what photoredox chemistry is good at. And so we were able to then implement almost exactly the same type of reaction, but starting from these silicon reagents instead of the tin. And to do this, we have a photocatalyst. We shine light on the photocatalyst to form the excited photocatalyst. This can now affect single electron oxidation of the carbon silicon bond. It forms the same type of end-centered radical. This undergoes cyclization, again, the 6-endo-trig cyclization to give an end-centered radical. And now the reduced photocatalyst does a single electron reduction in order to deliver the saturated N heterocycle and the ground state photocatalyst. So now we have a way of generating these different type of SLAP reagents, and by using light and a photocatalyst, we can produce these saturated N heterocycles using light as the key reagent. So we developed this approach as a plate-based synthesis, and we worked in collaboration with a platform at Novartis in Basel, so what they call the microcycle platform, where they have invested in some synthetic automation. We actually did most of this work in our lab at ETH, but we really benefited from Novartis's experience, particularly in some really incredible automated analysis and purification that I will show you in a minute. So we were able to run hundreds of reactions at ETH using a photoreactor, transfer those to Novartis, where they would do purification and isolation of those, and then some robotic-based plating out of the different compounds. And then from those purified compounds, they could determine the physiochemical properties. We could look at some basic bioassays and things like solubility and permeability to give us information about these compounds. And along the way, we also would learn a lot about what works and what does not work, so that back in our lab at ETH, we could use this information to develop a machine learning approach to predict which combinations of reagents would be successful and which ones would not. So for the parallel synthesis, so we ended up basically implementing this in 96 well plates that look something like this. And we developed a couple different types of reagents that we can combine from a starting aldehyde or ketone in combination with the sulfonyl imine. And in a fully automated process, we can generate this library of SLAP reagents. And then we use a small robot to transfer those out, and then add a different aldehyde or ketone, followed by the photochemical cyclization reaction in order to produce the saturated N heterocycles. We developed special plate-based photoreactors for doing this. So what we did, Moritz and Benedict did, is we bought these individual LED lamps. So we have one LED lamp per reaction, and then we implemented this in a water-cooled approach. So we use pretty high-powered LED lamps here, and they're spaced, quite closely together because we're trying to do these on these commercial 96 well plates. And as you can imagine, this gets quite hot, so we have to be a bit careful about how we do this. But with the help of our machine shop at ETH, we could build this jacketed reaction and hook this up to a cooling system. And this allows us to conduct the photochemistry while maintaining a constant internal temperature of about 30 to 40 degrees Celsius, despite the high intensity light that we use. So once all this was established, we could develop a system where we can produce multiple plates. And so we use multi-channel pipetters to start with the aldehydes. We have different of the SLAP reagents that we prepare by using this bifunctional reagent, the starting aldehyde. So we make the sulfonyl imine, we do a radical addition to that, and then removal of the sulfonyl group, followed by filtration. We have special plates for filtration, and this gives us the SLAP reagents, and then we take those with the aldehydes, a variety of different aldehydes to form the imines. And then we take those imines and add a photocatalyst and use the photoreactor to produce the saturated N heterocycles. And we then take those, dilute them, and at ETH, we look at the reaction profiles by LCMS. And then we transfer these plates to Novartis, where they can isolate the compounds and examine their physiochemical properties. And this works actually quite well. So we've made almost 1,000 compounds by doing this. So there are 6 plates that are shown on this slide here. And this is a kind of heat map. I apologize for the colors. They maybe look a bit unusual. I'm colorblind. So this is the solution that my group came up with so that I can actually read one of these. And what you see from these kind of heat maps is that not everything works. We have kind of islands where certain reagents do not work. But overall, most of the reactions give at least a little bit of product. So out of 576 reactions that are shown on this slide here, we were able to detect product from 461 of them. So that's a success rate of at least finding some product of about 80%. The cases where it doesn't fail, where it fails, are sometimes we have reagents that are too hindered. Sometimes we have some strange heteroaromatic compounds that interfere with the reaction. But I would say by and large, it works really quite well. And what was really great about working with Novartis is that they actually had built this almost fully automated approach to actually purify so many different reactions by preparative LCMS. So they have a system that takes the crude materials that we produced at ETH. They have an analytical UPLC that will measure them and use a charged aerosol detector to see even non-UV active products. They can then basically transfer this in an almost automated way, develop conditions for this, and a robotic system that will then purify all of these different compounds. And so they took all 576 of these reactions, or at least the ones that worked. And from that, they were able to isolate 307 different saturated N heterocycles. A few of them are shown here. And in a few cases, in about 70 cases, we were actually able to observe the formation of diastereomers and even separate those diastereomers. And from those isolated purified compounds, we could look at different physiochemical properties. So we could look, for example, about the pKa of the molecules. These are all amines, and so what we see is a kind of the expected profile for a basic compound. And the colors here represent different sets, so morpholine or piperazines or things like this, alkyl or aromatic residues. And so we see the range of physiochemical properties. We can also look at things like solubility. And here, by having so many different compounds in the same series, we can actually look at some trends. And very interestingly, and I have to say a little bit surprisingly, what we find is that certain compounds, so for example, morpholines that have at least one aliphatic group show very good solubility. And the same is true of the piperazines, where we have at least one aliphatic group. In contrast, things like these dialkyl piperidines, which we would have expected to be more soluble, are maybe not as soluble as we would have expected. And so already, even from these few hundred compounds, we're learning some things that we would not have been easily able to predict by just looking at the molecules. And likewise, this is just something I found interesting. If we look at all of the cases where we could isolate the diastereomers, in some cases, the diastereomers have very similar properties, But in many cases, we actually see quite a bit of difference in the log D or the solubility of the different diastereomers. And so this is also something that I would not really have predicted had we not actually produced the compounds and measured them. So now that we have this pretty good and semi-automated way of preparing these saturated N heterocycles on plates, we can go back and again, look at the commercial libraries of aldehydes and ketones, for example, from enamine. And we can say, okay, if we start from these commercially available aldehydes, we combine them together with our bifunctional reagent, and then take another aldehyde and do a SLAP reaction. These are the types of products that we can form. So if we start from enamine stock aldehyde and ketones, we can combine those together. We can make a very large virtual library. We can filter out certain products that we don't want to use, and then take the remaining ones and enumerate them into a virtual library with about 20 million compounds. So now we have this virtual library we can screen. And if we get a hit, then we will say, okay, now I want to produce, these 20 morpholines or piperazines or oxazopanes or whatever we find from that virtual screen. And I think here's where, the interesting question arises. So, we want to have a vending machine. We want to say, okay, here's a menu that you can choose from. Our menu's pretty big, so maybe 20 million compounds. But of course, it's organic chemistry, so not everything is going to work. And I think for a lot of chemists, the question is this really a vending machine where I type in what I want, and as long as it's offered by the vending machine, I'm going to get it? Or is this going to be more like gambling, more like a slot machine where sometimes I get it and sometimes not? And until I actually try, I'm not going to be sure which one it is. And so here, what we want to do is we want to take the data that we have, these 500-600 reactions that we have already done, and see if we can use that information to build an algorithm or a machine learning based approach that will predict whether or not our combinations of building blocks are going to be successful. So we can take our virtual library and we can use different types of representations. So here's one type of representation. This is not exactly chemical space. This is each dot represents a representative member of the library. And the distance between the dots is a difference between their similarity as measured by a number of different parameters. And so the red dots are ones that we actually synthesized. So you can see that from our training set or molecules that we've actually made, we don't cover the full virtual library, but it's maybe not so bad. I would say that we have decent coverage, except maybe for some islands here. And then eventually we also want to make a validation set. So we would buy some aldehydes or some ketones that we've never tried before and see whether or not we're going to be able to make those. But the first step is to try to build an algorithm that will predict whether or not we're going to be successful in performing the SLAP chemistry. So we take the products that we would like to make and we can encode them in a graph. And we can then annotate the properties of this in a variety of different ways, such as by using RDKit to enumerate the properties. I have to say at this stage that this is really, I'm not an expert. I have very good people in our group, such as Julian Goetz, who's a PhD student, who really works on this. And so if you have detailed questions about the machine learning parts, I'm afraid I'm not going to be able to answer them, but we can get back to you. And so what we are doing now is basically we're building a machine learning model where we take our known data. So these are actual results. We know what our starting materials are. And then we have cases where we know what one of the reactants is and not the other one. And then we have cases where we the machine learning model has not seen either of the reactants. So if it's seen one of them, we call this a 1D out of sample. If it's never seen either of the reagents, it has to predict the properties of everything. We call that a 2D out of sample. And if it has seen somewhere in its training set both of the reactants, then we call that in sample. And we use tried a variety of different methods to then see if we can predict which reagent combinations are going to be successful and which ones are not. And so what we can see here is that for in-sample ones where the algorithm has seen both of the reagents before, we can get about a 80–85% success rate in terms of predicting whether or not the reaction will be successful or not. But if it is completely out of sample, it's never seen either reagent, and it has to use the chemical information from what it knows to predict, it gets it right about 66% of the time. This is not too bad. It's not perfect. And it allows us to basically then take our library and for any given product that we would like to make, we can predict with not a high, but not so terrible level of accuracy about whether or not that reaction is going to be successful. Now, admittedly, I think our training set, even though it is very big for what other organic chemists have done, so a few hundred molecules, so maybe about 500 molecules or 500 reactions, for an actual machine learning based system, this is still pathetically small. So we're quite interested in how we use automation and other types of chemistry to build much larger data sets that will be able to make more accurate predictions. But already now with this medium-sized or data set, we can start to get some level of accuracy in terms of predicting what will work and what does not. And now what we're doing is we go back to enamine, and we have sort of randomly picked a bunch of molecules and run those reactions so we can see which ones work and which ones don't. And then see how well we're able to predict the reaction outcomes. So that's ongoing work right now. And what we've learned along the way is that in order to make these kind of predictions, so in the end, if we're going to be able to say for our virtual library, yes, you can make this particular member, or this one might be difficult, is that we really need a lot of data, and generating the data is a bottleneck. So we have other chemistry now where we've run 50,000 reactions. We are working on analyzing them. We build a lot of automated data processing systems to identify which reactions are working, which ones are not, what the byproducts are, what is going on. And so we're trying to build a very large data set that can be a prediction or hopefully a more accurate prediction of which combinations will work and which ones will not. And that's going quite well, but of course it will be self-contained. So it will make predictions, but only within the realm of that particular reaction. And that gets into another point, is that if you want to have this kind of data, which ultimately we need, we really need automation. Obviously, we're not going to be able to make, even 100 reactions is difficult. 1,000 reactions is almost the limit of what can be done. And we really want to get to the point where we can run more reliably 10,000s, eventually 100,000 different reactions. And so we need not only the automation approach, we need the analytics, we need the chemoinformatics in order to be able to do all of this kind of analysis in an automated way. But we also need the right types of reactions that are suitable for this. So we have a few that we've developed in our lab, but I think developing more, particularly 3-component couplings, particularly things that start from aldehydes and amines, where we can get a good supply of building blocks, are going to be very important. And I think the learnings from this is, again, what nature has taught us, that if you really want to make a lot of molecules and do it reliably, we need to start from interesting preformed building blocks. So nature uses amino acids, and I think that's a great starting point. For some of our work, we use things like the SNAP reagents and also some more exotic starting materials that we've developed in our group for doing this very interesting assembly chemistry. But it always shifts a little bit the problem to we have to be able to produce these kind of starting materials. And in many cases, it's worth it. But as much as possible, we'd like to work from the materials that we can buy easily, like amines, acids, and aldehydes. Okay, so I think we'll stop there. And again, thank my research group for really diverse and dynamic research group who works on many different kinds of problems. So we do a lot of work also in chemical biology, and peptides and proteins, and always something new and different going on. I really admire all of them for being fearless and going off and doing something that is completely different maybe than what we know. It makes it very interesting and exciting to come to work and talk to them. And for this talk, I really need to thank a few of the PhD students. So Moritz and Ice both graduated a while ago. Julian and Angus are still PhD students in the group, and they have developed a lot of the chemistry, and Julian has worked on the machine learning part, and Julian and Angus and others have worked also on the virtual libraries and the enumeration of the virtual libraries, so that we can build these and then eventually really prove whether or not we can make them or not. And we've had a great collaboration with the microcycle platform at Novartis, led by Kara Brocklehurst and assisted by many talented chemists who really did an incredible job purifying hundreds of compounds, running quantitative NMRs on many different compounds, and measuring the properties of the compounds, so we can really learn something from that effort. And yeah, I thank you again all for attending, and really for the invitation and the honor to present, and I'd be happy to answer any questions.
Thank you very much for a very interesting and impressive presentation. This is the first time that the automation of organic synthesis on our seminar. Dear colleagues, do you have questions to Jeffrey? I don't see any hands.
Yes.
Oh, okay, good.
I can start if you. I've just got the lightning here, so you can see me. It was blackout for a while. So Jeffrey, can we formulate now a simple principle to compare reactions? So, for example, one reaction is better than the other to be suited for automated synthesis in general, and for your technology, first of all.
Yeah, I think that's a really good question. Let me see if I can. So I think if you look at reactions that are good for automation, let me see. So not every reaction is well suited for automation. So of course you want something that has a relatively standard reaction conditions. So this was the advantage of SNAP chemistry, because basically all the different SNAP reagents work under a quite standard set of reaction conditions. That may not be the very best reaction conditions, but we have conditions that will deliver product in most cases. And I think that's true of things like reductive amination. We've had very good luck. Actually, this worked better than expected in the reductive amination. Amide bond formation is not quite as good, but it, as long as it's not too hindered, we can have a solid supported coupling reagent that works most of the time. And so, for many standard things, it works quite well. We're doing now Suzuki couplings, and I have to say we in the end, we have a system that works not too bad. It will do quite a few things, but Suzuki is not as robust. So if you have a lot of heteroatoms or something, you might need some fancy ligand, or you might need a very special palladium precatalyst. And that's much more difficult for us to automate. But I think chemists understand that. So if you do a reductive amination, you sort of expect it will work. And so you have maybe, you will be disappointed if you try it on our machine and it doesn't work, but this does happen. With Suzuki coupling, I think what happens is you will take our capsule and you will try it, and if it works, you're happy. But if it doesn't work, you understand that this is a reaction that might be a bit more picky, and you're not always going to get as lucky that your particular substrates work on that. I think perhaps the more interesting question is, how many reactions do we need? So there are so many reactions nowadays. And if you're doing medchem, if you're really trying to go and fine tune your molecule, then you want some really exotic reactions, very special reactions. And those are always going to be important. We're always going to need this last fine tuning. I think what we're interested in is saying, okay, we can't do everything, but here are 10 or 15 reactions that we can do reliably in an automated format. And we can go and look for these lead structures. So we can say, yes, I can make this molecule, I can make it quickly, I can make it in 3-4 days, and I can test it. And then that will give me the information that I need to say I'm on the right track, or no, this is, the computer is just completely wrong in what it's predicting. Until we actually have a molecule in hand, we can't make that decision. And what we want to do is to make it as painless and quick as possible for a chemist to produce a molecule, to get an assay result, and then be able to go on with the process.
Thank you. Fantastic talk. Thank you very much.
Thank you.
Yes. And you answered one of my questions, by the way, about how many reactions we need. Other question, how long does it take to optimize one reaction for automatization?
That's a very good question. So I would say it takes right now, if we have one chemist working on it, it takes about 3 or 4 months to optimize a reaction. So, some are easier than others. Suzuki maybe takes longer than Bode protection, but it does take some time to do this. And our standards are pretty high, right? I mean, this is, and this is done now, not so much by my group, but by the company. This is a commercial product. They're selling it. They want, if they're going to sell it and have customers, the reaction needs to be relatively reliable. So they will probably put more effort into the optimization than we might if we're just publishing a paper. So there's a lot of little details that we think about. So what is the right support? What is the right concentration? What is the right amount? What is the solvent compatibility? And of course, we always want a trade-off between the most general reaction conditions possible with ones that might work on particularly different substrates. And so we pay a lot of attention to that. We like to work with certain solvents and things like this. We want to avoid having to cool the reaction because the machines aren't good at cooling. They're pretty good at heating. So we try to find conditions that are most suitable for that. We also want the capsules to be stable. So once we package them, we want to make sure that you can keep it for months or years without it decomposing in the meantime. So sometimes we have to make some compromises between what is the absolute best reagent and what is a stable formulation.
Thank you. Your colleagues? Other question? I have also other question to you about, is it really a real-time flow process, or you use flow and batch combination?
Yes, exactly. It's a combination of flow and batch. So basically what happens is we start from our, for example, aldehyde. The machine adds solvent, and then it sucks up the dissolved aldehyde. It passes it through one component, and recirculates it. So we can tell it, okay, recirculate it for 10 minutes or however many times. And this is just a line in the program. And then we tell it to change the valve and take it and circulate it through the other component of the capsule and so on. So it is definitely a combination between flow and batch. Yeah.
And then the next question, what advantages or disadvantages of your method and flow chemistry? It's a both method of optimization of synthesis.
Right. So I think flow chemistry is really great when you have an optimized process. So let's say you have something that you want to produce on scale and you optimize the flow reaction, then you can set it up. You can have a apparatus which is set up and you turn it on and you let it go and it produces molecule. And when you have enough, you turn it off. And then when you need more, you can easily go back and do this again. So you put a lot of optimization into, the flow system for that particular chemical reaction, and then you have a very reliable way of producing it. Our system is more for making a small amount of different types of compounds. So we have one system, but because it is this combination of flow and batch, we can make like 1 millimole at a time. So it's a bit like the coffee makers, right? So if you want 2 coffees, you need 2 capsules. It's the same here. If you want 2 millimoles of products, you need to use, run it once with 1 capsule and then get another capsule and make another millimole. So our system is really for prototyping. It's for making small amounts, and flow chemistry is for scaling up something that you want to make more of.
Okay, I understand. Okay, Professor Lipson, do you have a question?
Yes, I have a question. Dear Jeffrey, could you tell us about what kind of reaction is most difficult to optimize for this automatic synthesis?
Yeah, so I would say anything that needs low temperature is very difficult to optimize. So we would have to build the cooling into the machine. This is difficult. And then, of course, air sensitive as we can deal with, but things that work at low temperature, then you start to run into solubility problems. Even with enolates or things like this, they often precipitate from the reaction. And so I think anything that needs -20 or lower temperature is going to be quite difficult to put on a flexible system like this.
And what amount of target compounds could you obtain in this synthesis?
So we typically target being able to make about 50 to 100 milligrams of material on this. This depends a little bit. The SNAP chemistry is actually quite interesting because since we're releasing the reagent from the resin, you can add a smaller amount or a larger amount, and it will always deliver the right amount of product. You don't even need to weigh it. You just need not to be over too much a maximum amount. For things like the reductive amination, there we have a fixed amount of reagent. So let's say we can go up to 2 millimoles. You can run with 0.5 millimole or 1 millimole, it doesn't care, but you can't go beyond the amount of stoichiometric reagent you have in the capsule. So we've targeted this to make basically 20 to 50 milligrams of product. So if you get 50 to 70% yield, we get between 20 to 50 milligrams of product, which is what we think med chemists want. For screening, people often want smaller amounts, and then that's why we go to these plate-based compound like formats for making tiny amounts of compounds that we might just do a quick assay on.
Thank you very much for your very interesting presentation. Thank you.
Thank you very much.
Oh, thanks. Dear colleagues, do you have more questions? I don't see. Okay. And do you have reviews in this field?
We have some, yes, I can send you some. We have a paper that's just should come out very soon about the first part of the talk. And I think we do have some reviews about the automation. The last part of the talk is quite new. We haven't published this yet. So that's not there yet, but we hope to submit that soon. Yeah.
Because it's very interesting to use it if it's possible for a lecture for our master students and PhD students, because I lecture modern synthesis in university, and of course your topic is modern synthesis, and I would like to make it a part of my lectures.
Okay,
for
that's
students.
very kind. Yeah, I will send you a few,
Okay,
a
thank
few
you.
reviews on the automation. Yeah.
Thank you very much. So, dear colleagues, if you don't have any question, let me thank again for your very nice presentation, your nice lecture, and wish you a nice day.
Thank you very much. Bye-bye.
Thank you. Bye.
Thank you.
Bye.